Is it good? Okay, thank you. In this slide, we are going to talk about reabsorption rate. Dear student, pay attention, please. We are behind our schedule, okay? Cooperate with me in order to go ahead. Regarding reabsorption. In proximity, reabsorption of water and solid. Okay. 65% of water and 65% of solid are reabsorbed. What does it mean? Suppose that when we say something is, be careful please, okay? When we say something is really filtered, it means it is filtered as easily as water. Got it? When we say something is really filtered, okay, it means it is filtered as easily as water. Okay, what does it mean? It means actually substances pass through simple diffusion, through osmosis. Okay, it means, for example, when sodium concentration in the blood glomerular capillary is 142 milliequivalent per milliliter. Okay, here inside the Bowman's capsule, the concentration of sodium is the same. Okay, it means the concentration of potassium is the same. Okay, here you can see 65% of water and 65% of solid are reabsorbed in proximal tubule. What does it mean? It means the concentration is the same in proximal tubule too. Because 65% of water and sodium are reabsorbed together. I mean osmolarity. Osmolarity means concentration of solids and water. Osmolarity in proximal tubule is the same as that in Bowman's capsule. Because as you know, sodium and water are reabsorbed together in the same amount. Was it clear? Yes. Very good. What happens in thin descending limb? Thin descending limb of loop of Henle. Don't forget, we don't have active transport mechanism for sodium reabsorption. There is no active reabsorption for sodium. Do we have permeability to sodium? What is the difference between permeability of membrane to sodium and active mechanism? Permeability means simple diffusion. Okay, yes, we have permeability. Permeability, yes, we have. Not in this part. We have permeability. It means sodium can pass through simple diffusion, not by active transport. Here, there is no active transport for sodium, but by 20% of water is reabsorbed. Okay, actually, let me draw a nephron. Bowman's capsule, proximal tubule, loop of Henle, distal tubule, collecting duct. Proximal tubule, thin descending limb, thin ascending limb, thick ascending limb, early distal tubule, late distal tubule, cortical collecting tubule, medially collecting tubule. Okay. Here, as I told you, 65% of, here, 65% of water solute are reabsorbed. What happens here? Actually, just 25% of water is reabsorbed. But from this part on, actually there is no permeability to water. But water is not reabsorbed from this part on. Even in the presence of ADH. Got it? Suppose that there is no ADH. There is no ADH. Which parts of nephron are permeable to water? Which parts? Proximal tubule and in descending limb. Got it? Suppose that there is, actually we have ADH. Which parts of nephron are permeable to water? Actually, proximal tubule and the descending limb are not sensitive to ADH. All the time they are permeable to water. Got it? How about this part? Suppose that we have ADH. Actually, this part is not permeable to water. I mean, the ascending limb is not permeable. We have ADH, okay? How about thick ascending limb? Thick ascending limb is not permeable to water. Why? Because it is not sensitive to ADH. How about early distal tubule? Early distal tubule. Actually, it is not permeable to water. In the presence of ADH. How about late distal tubule? Don't forget, from late distal tubule on, actually, late distal tubule, cortical collecting tubule, medullary collecting tubule, actually respond to ADH. Got it? These are these parts which increase permeability to water when there is water deficiency. Why do I say water deficiency? When there is water deficiency, ADH is released. Got it? Very good, don't forget. Okay. Okay. Don't you have any question? No question? Let me continue. Reabsorption. Right. In the ascending limb of loop of head, do we have water reabsorption?
No.
No, never. Even in the presence of ADH. Do we have active mechanisms for sodium reabsorption? No. In this part, there is no active mechanism. But we have permeability. It is passive. Very good. Got it? How about thick ascending limb? Do we have water permeability? Do we have active mechanism for sodium? Yes. Actually, 25% of sodium is reabsorbed in thick ascending limb. For this reason, we say thick. When we say thick, it means cells are great, are big. Got it? And in distal tubule, actually, early distal tubule, do we have water reabsorption? No. And solute, yes, actually 5% of solute are reabsorbed in distal tubule. Early and late distal tubule together. Got it?
3%.
Yes. Actually, reabsorption in the other parts of nephron depends on our needs of our body. Got it? Based on the needs, solute and water are reabsorbed. Got it? No question. Reabsorption in proximal tubule. I told you 65% of water and 65% of solute are reabsorbed. By which mechanism? Actually, you can see one epithelial cells in proximal tubule. Don't forget, we have sodium glucose transporter, SGLT. Do you remember SGLT from cell physiology? SGLT is secondary active transport. And sodium amino acids transporter or co-transporter. It is another example of secondary active transport. Ready? Don't forget, glucose and amino acid are reabsorbed just in proximal tubule, not in the other parts of the nephron. Why? Because we don't have any transporter, a specialized transporter for glucose and amino acid reabsorption. Got it? Suppose that in a person is suffering from diabetes mellitus. What happens? Actually, glucose concentration here is high. Okay? These transporters cannot reabsorb all glucose. What happens? Glucose is not reabsorbed in the other parts of the nephron. For this reason, glucose is actually appears inside the urine. But don't forget this transporter and this one are found just in proximal tubule. Okay, what's the fate of these actually substances inside the cell? Glucose is reabsorbed by GLUT. GLUT is facilitated diffusion, simple diffusion, active transport. Which one? Active.
Facilitated diffusion.
Facilitated. When you say GLUT, GLUT means glucose transporter. It is facilitated diffusion. When we say SGLT, sodium glucose transporter,
Secondary
don't
active
forget,
transport.
it refers to secondary active transport. Got it? Actually, and what's the cause of reabsorption in this luminal membrane? What's the main cause of reabsorption? Don't forget,
Sodium.
existence of sodium-potassium pump or sodium-potassium ATPase is the main cause for reabsorption. Okay, we have talked about this part in the previous session. And amino acids are reabsorbed in different actual mechanisms. It shows here simple diffusion. Got it? No question? Do we have separation in proximal tubule? Here you can see just reabsorption. Do we have secretion? Yes, of course. Here. Again, existence of sodium-potassium pump in the basolateral, not in the luminal membrane. In the basolateral membrane process, sodium is reabsorbed. Okay? Sometimes this reabsorption is coupled with the reabsorption of something else or secretion of something else. Here you can see sodium, hydrogen secretion. Don't forget, everywhere you see hydrogen secretion. What does it mean? It means
Active with the
it is important in acid-base regulation.
bicarbonate.
It is secreted hydrogen. It is the subject of the last sessions. Okay? It is secreted in order to reabsorb bicarbonate. Okay? Keep it in your mind. We will talk about that. Hydrogen is secreted in order to reabsorb actually bicarbonate. But we will talk about that. If I ask you regarding the secretion in proximal tubule, you have to answer yes, we have secretion. For example, sodium-hydrogen exchanger, sodium-hydrogen antiport, sodium-hydrogen counter-transport. They are the same, okay? And by which mechanism? Simple diffusion, facilitated diffusion, or primary or secondary active transport?
Simple diffusion.
Simple. Is it simple diffusion?
Hydrogen.
Secondary active transport. When we say antiport, counter-transport, exchanger, or co-transport, symport or secondary active transport. Okay? Don't forget. Got it? If we have enough time, I'll try to make it easy for you. Okay? I try to talk about different transport. Actually, do you want me to talk about different active transport, primary, secondary? Do you need?
Yes.
Let me talk about that. It is very important. Okay, be careful. Transport mechanism extracellular. I describe it, and after that you have to guess which transport mechanism, for example, it is. Got it? Okay. First one is, don't type this. Simple diffusion. Simple diffusion is done through channels. If you hear channel, okay, any type of channel, it will be simple diffusion. Okay? Channels might be lead channels. They have no gate. All the time they are open, okay? Or gated channels. Okay? Gated channels might be voltage gated channels. Actually, they are involved in action and potential. Okay? Ligand gated channels. Ligand gated channels have binding site for something. After binding, for example, an acetylcholine, what happens? The gate of channel is open and allow substances to actually move based on simple diffusion. When we say channel, channel is for simple diffusion. Don't forget, got it? Or stretch gated channel, stretch or pressure gated channels, okay? All of these channels actually simple diffusion. Got it? Lead channel, ligand gated channel, voltage gated channel, stretch or pressure gated channel. Don't forget, got it? And we have carriers too. These are channels. We have two types of carriers. Carriers which are involved in facilitated diffusion. When we say diffusion means we don't need energy as ATP, okay? Either in facilitated or in simple diffusion. Carriers which are involved in active transport, okay? Active transport. Facilitated diffusion. In facilitated diffusion, we have a channel, we have a protein. It is not channel, okay? You cannot see any pore. Okay, but in the channel you can see a pore, okay? In carriers, in all of the carriers, all of them, either in facilitated or active, we have a binding site for something. After binding, for example, after binding glucose, it is glucose transporter. After binding glucose, there is a conformational change in the shape of this carrier, and glucose is released in the opposite side of the membrane. When we say GLUT is facilitated diffusion for glucose. Got it? Regarding active transport, we have two types of active transport, primary and secondary. Primary has a sign. When we say ATPase or pump, at the end of anything, it is primary active transport. Potassium, hydrogen, ATPase. Sodium, potassium, ATPase, or sodium, potassium, pump. When we say pump or ATPase, at the end of anything, it is primary active transport. Got it? How about secondary active transport? Primary might be, for example, for one component such as hydrogen, ATPase, but secondary actually, or might be for two substances, for example, sodium, potassium pump. Got it? How about secondary? Secondary, actually two substances actually are transported. Okay? If the transporters, actually the direction are in the is the same, is in the one direction, we say it is co-transport or symport. Okay? I say sodium glucose co-transporter. Sodium glucose symport. Okay? It is secondary active transport, and it means both are transporting in one direction. Got it? Sometimes they are in the opposite direction. For example, we say sodium hydrogen exchanger. sodium hydrogen countertransport, sodium hydrogen antiport. They are the same. Okay, these are secondary active transports. Secondary might be done in one direction, might be in the opposite direction. No question. Got it? Now I want you to actually guess the mechanism. Sodium hydrogen exchanger or antiport or countertransport. Secondary active transport. Very good. Very good. And how about this one? Sodium potassium ATP. Primary active transport. Got it? Okay. We are in proximal tubule. Okay. We have talked about reabsorption and secretion in proximal tubule. Do you have any question? No? Okay. Pinocytosis. Actually, I told you proteins are not filtered in Bowman's capsule. But it is not correct. Actually, little amount of proteins are filtered in Bowman's capsule. And this amount of protein must be reabsorbed. The absorption of protein is done by pinocytosis. Actually, endocytosis is done in two ways. One is pinocytosis, and the second one is phagocytosis. Phagocytosis is for white blood cells. Pinocytosis is for micromolecules such as proteins. Okay, it is endocytosis. Is it need energy? Does it need energy? Yes, as ATP. Endocytosis, exocytosis, and active transport needs energy as ATP. All actual transport mechanisms are are done without energy, but these three mechanisms need energy as ATP. Okay, mechanism, we are in proximal tubule. Mechanisms by which water, chloride, and urea reabsorption are coupled with sodium reabsorption. When sodium is reabsorbed by sodium glucose transporter, sodium hydrogen exchanger, sodium amino acid transporter, as I told you, what happens actually, water is reabsorbed. Why? Because all the time water follows sodium. Water is reabsorbed based on osmosis. When sodium is reabsorbed. What happens? Suppose that sodium is reabsorbed, water is reabsorbed. What happens? We have increase in concentration of urea and chloride ions. For this reason, we have increase in concentration. Okay, what happens? We have passive urea reabsorption and passive chloride reabsorption. Passive means simple diffusion by simple diffusion. Okay. On the other hand, when sodium is reabsorbed, positively charged ions are actually reabsorbing or removed from tubules. What happens? We have excess negativity, excess negative charges. For this reason, this negativity causes chloride ion to be reabsorbed more. But after sodium and water reabsorption, we have chloride and urea reabsorption in proximal tubule. Okay, in the other parts of nephron, actually solute or water are not allowed to be reabsorbed in this way. Got it? Okay. Actually, when you see these cells, it means we have active transport mechanisms. For example, in proximal cells, you can see brush borders, and these cells, a lot of mitochondria. It means we have active transport mechanisms. Okay, but here you can see cells are thin in thin descending limb and thin ascending limb, and they don't have mitochondria. It means the absorption is done based on actually simple diffusion. Okay, doesn't need any energy. As I told you, 65% of water and solute are reabsorbed in proximal tubule. You can see the substances which are secreted into the proximal tubule. Actually, one substance which is secreted is hydrogen. We have talked about it. Okay, and you can see organic acids and bases actually are secreted by salts, oxalate, urate, catecholamines, some drugs, diuretics, and PAH. You are familiar with PAH, para amino hippuric acid. Keep it in your mind. We are gonna talk about PAH in renal blood flow measurement. Got it? It is very important, okay? This axis shows, this axis shows a fraction, concentration of a substance in tubules, tubular fluid, into the concentration of a substance in plasma. Here you can see the first part of proximal tubule, and here is the end of proximal tubule. Got it? Okay. I told you sodium concentration here is the same as plasma. Am I right? How about in proximal tubule? In proximal tubule, 65% of water, 65% of sodium are reabsorbed. What does it mean? It means the concentration of sodium here remains the same, 142. Got it? No question? Okay. Now I want to get this fraction for sodium ions. For sodium ions, 142 in tubules in proximal tubule to 142 in plasma. The fraction will be 1. It means the concentration of sodium remains the same. Okay? From Bowman's capsule to the end of proximal tubule. Yes, that's right. You can see 1 for sodium ions. How about water? Let me say osmolarity, it means for water. Actually, for water, it is 1. Why? Because 65% of water are reabsorbed. Got it? No question? And how about chloride ions? Concentration of chloride ions in plasma is the same as at the end of proximal tubule. Got it? For this reason, for chloride it will be 1. Okay. Do you remember I told you 100% of glucose and amino acids are reabsorbed? Do you remember that? 100%. All glucose and amino acids are reabsorbed in proximal tubule. Do you remember? Yes. Alhamdulillah. What does it mean? It means concentration of glucose at the end of proximal tubule is 0. And how about concentration in blood? For example, it might be, 100 milligram per deciliter. Okay? And it will be 0. How about amino acid? It will be 0. You can see the fraction decreases for glucose and amino acids. Okay? Until it actually reaches 0. Okay? At the end of proximal tubule. When you see something is less than 1, it means here we have reabsorption more than water and sodium. Got it? Because here 65% of sodium is reabsorbed, but 100% of glucose and amino acids are reabsorbed. Okay, below actually the line of 1, it means reabsorption is more than sodium and water, is more than 65%. Dear daughters, don't you have any problem? No? How about bicarbonate? What do you think? Reabsorption is more than 65%. Yes, it is something about 80 or 85% of bicarbonate are reabsorbed. Okay?
Should reabsorb. How reabsorb?
we will talk about in acid-base regulation.
The luminal border of the proximal crude tube don't pass with this molecule.
Which molecule?
Uh,
Bicarbonate, for
yes.
example. we
You
will
should
talk about that.
you should active with the hydrogen.
Yes, yes. After binding to hydrogen, we have carbonic acid. Carbonic acid is converted to carbon dioxide and water. We easily, they only absorb. We will talk about that, okay? No question. Actually, 100% of glucose and amino acids are reabsorbed. 80 to 85% of bicarbonate are reabsorbed in proximal tube. Yes.
Can you repeat and I inhale for absorption?
Yes. 65% of water. Suppose that I have a solution. Okay. I took the same volume of sodium and water together. Okay. It means the concentration of sodium and water remain actually constant. For example, in this glass, in this container, and in actually the volume that I have taken. Got it? For this reason, it remains 1. Got it? No question? Dear Manoj, no question? How about urea and creatine? Do you remember I told you we have little amount of actual reabsorption for urea? Do you remember that? But it is reabsorbed less than water and sodium. For this reason, tubular fluid concentration in tubules increases. For this reason, actually, the fraction will be more than 1. How about creatinine? For creatinine, there is no reabsorption. It remains inside the tubules and it becomes more concentrated. For this reason, concentration in tubular fluid is more than concentration in plasma. Got it? Over this, actually the line 1, it means actually reabsorption is less than sodium or there is no reabsorption. Below this line, reabsorption is more than sodium and water. No question? In the descending limb, what happens? Water is reabsorbed. 20% of water is reabsorbed in this part. From this part on there is no permeability to water. Okay, we don't have active mechanisms here, active transport mechanism in descending and ascending limb, but there is little permeability to sodium ions, sodium and chloride ions. Regarding thick ascending limb, actually, is it thick ascending limb? Does it reabsorb water?
Sodium.
Just sodium solutes, 25% of solute are reabsorbed here. Which solutes, for example, sodium, chloride, potassium, calcium, bicarbonate, magnesium, and do they have secretion? Yes. Wherever we see hydrogen secretion, it means actually it wants to reabsorb bicarbonate.
Yes.
Why? Because bicarbonate, it is essential for our body. It is a buffer system in our body. We will talk about that. Got it? Regarding transporters in thick ascending limb, which mechanisms are seen in basolateral membrane? Sodium, potassium pump. It is primary active transport. Chloride channels, single diffusion. It is single diffusion. Potassium channels, single diffusion. And what happens here? In luminal membrane, we have sodium, potassium, 2 chloride transporters. Sodium, potassium, 2 chloride, symport or co-transporter. It is secondary. Actually, 4 components are transported by these actually proteins: sodium, 2 chloride, and potassium. If I ask you in which part of nephron sodium potassium 2 chloride transporter is seen in which part? Big ascending limb, don't forget. And some drugs actually inhibits this transporter, such as furosemide. It is used in hypertensive people, hypertensive patients, okay? Actually, as a result of these drugs, this transporter, this co-transporter is inhibited. Sodium is not reabsorbed, for this reason water is not reabsorbed, and what happens?
Accumulate.
Actually, water is excreted into the urine and blood volume decreases, blood pressure decreases. Got it? And do we have secretion? Yes, sodium hydrogen secretion, sodium hydrogen exchanger, sodium hydrogen antiport or counter transport. It is secondary active transport. Very good. Don't you have any questions? Dear Manor, dear Fsلجة, what's the Shabab? I forgot it. Okay. Dear Shabab, don't forget, in peak ascending limb, sodium, potassium, magnesium, and calcium are reabsorbed from actually paracellular pathways. But water is not reabsorbed. But we will talk about that. Early distal tubule. Who can talk about mechanisms in early distal tubule? Actually, the main cause of reabsorption in this part is sodium, potassium, pump, primary active transport. And what's the name of this transport? Sodium chloride co-transporter or sodium chloride transporter. It is secondary. Very good. And it is chloride channel, simple diffusion. Very good. Don't forget, thiazide diuretics inhibits actually this transporter. It is used in hypertensive people too. Got it?
Yes.
And as I told you, 5% of filtered load of sodium chloride is reabsorbed in early distal tubule. Okay. We are going to talk about late distal tubules and cortical collecting tubule together because the mechanisms are the same. Okay. Actually, in late distal tubules and cortical collecting tubules, we have two types of cells: principal cells, intercalated cells. Principal cells, intercalated cells. When you say intercalated cells, don't forget, it is involving acid-base regulation, intercalated cells, okay? Here you can see principal cells. Which mechanism are seen? The main mechanism is this one. Sodium, potassium pump in basolateral membrane. I told you we never see sodium, potassium pump in luminal membrane, in the kidneys, okay? Sodium, potassium ATPase or pump in actually basolateral membrane is the main cause of reabsorption, okay? And here you can see sodium channels based on simple diffusion. And potassium channels. As you can see, potassium is secreted in these parts. And here you can see chloride channels. Okay. Some sodium channel blockers actually inhibits this protein. And sodium cannot reabsorb anymore. For example, such as amiloride. It is used in hypertensive people too. Okay, and aldosterone antagonists such as spironolactone inhibits principal cells. But if, for example, they inhibit sodium-potassium pump here, we will talk about aldosterone, and you can guess the mechanisms of aldosterone antagonists. Got it? No question? It is principal cell in late distal tubule and cortical collecting tubule. Got it? intercalated cells. Actually, you want to take a break? Some students are taking it now. After 10 minutes, I see.
15